I mean, I want you to imagine, just for a second, that you are standing in the middle of a high-latitude peat bog.
Oh, that is not a comfortable place to be.
No, definitely not. We aren't talking about, like, a pleasant, breezy meadow here.
Yeah.
This is a freezing, waterlogged, highly acidic environment.
Yeah, pretty much one of the most hostile places you can stand.
Right. The ground beneath you is basically this spongy, unstable muck. It's just a matrix of dead and dying plant matter. The air is damp, it's cold, it's heavy.
And for most forms of life on Earth, this is just an incredibly inhospitable place.
Exactly. It's essentially this nutrient-starved wasteland where the normal rules of decay just, well, they simply do not apply.
They really don't.
But if you look right down at your feet, right there, nestled into the damp, dripping sphagnum moss, you might actually see something remarkable.
Something that really shouldn't be surviving there.
Right. It's a highly specialized, statuesque little mushroom. It's called the Olive Brownie, or if you prefer the scientific name, Hypholoma myosotis.
And it really is just a striking visual when you see it out in the field. I mean, you have this tiny, almost architectural organism standing tall in an environment that actively resists decomposition.
And life as we typically understand it, honestly.
Yeah, exactly. The moss around it is literally engineering the soil chemistry to freeze time, and yet somehow this fungus has found a way to thrive.
So if you want to understand how a single, seemingly obscure organism can hold the keys to, the Earth's massive carbon vaults, you really have to look closely at this exact mushroom.
You do. It's crucial.
Today's deep dive is tracking a centuries-old scientific paradox centered around the olive brownie. We're looking at this massive stack of mycological, botanical, and ecological research to unpack, well, everything.
There is a lot to unpack.
There really is. We're getting into its bizarre chemical armor, its highly specialized structural engineering, and the profound ecological role it plays.
Because this tiny olive and bronze mushroom is essentially an environmental sentinel. It's operating right on the front lines of global climate mechanics.
Which is wild to think about.
Yeah.
And it also happens to be a species that has given taxonomists and evolutionary biologists a massive, sustained headache for, what, over 200 years?
Oh, easily 200 years. I mean, to really grasp the unique nature of this mushroom, we have to look at the historical identity crisis it has suffered.
Right, because the confusion begins right at its name, which is highly contradictory.
So contradictory. The mouse ear identity crisis, essentially.
Let's get into that.
Yeah.
The accepted scientific name right now is Hypholoma myosotis. And if we break down that species name, myosotis comes from classical Greek.
Right.
We have mus, which means mouse, and otis, or otos, which means ear, literally mouse ear.
Which is, well, it's strange for a fungus.
Yeah, it doesn't make any sense, but the plot thickens when you consider the botanical connection, right?
It does, because myosotis is the exact same genus name used for that famous delicate blue flower, the forget-me-not.
And botanists originally gave the flower that name because its short, pointed, sort of hairy leaves vaguely resemble the shape and texture of a mouse's ear.
Which makes sense for the flower. But the historical context here is critical. The olive brownie mushroom was first formally documented back in 1818.
By Elias Magnus Fries.
Exactly. Fries was a Swedish botanist, and he's widely considered the founding father of modern mushroom taxonomy. He placed it in this massive genus and called it Agaricus myosotis.
Okay, I have to pause here because the logic seems incredibly tenuous.
Yeah.
I mean, why would a preeminent 19th century botanist look at an olive bronze, slimy mushroom growing in a freezing swamp and name it after a bright blue flower?
Wow.
Which in turn was named after a rodent seer.
Yeah.
It feels like, I don't know, early astronomers misclassifying a distant galaxy as a localized gas nebula just because the lens of their telescope was blurry. Was Fries just looking at it wrong?
No, not exactly wrong. Just he was relying heavily on morphological parallels.
Yeah.
You know how things look to the naked eye at very specific stages of development?
Okay, so a specific stage.
Right. Fries likely observed the mushroom in its very early primordial stages. When the cap of the olive brownie is young, it hasn't flattened out yet. It's conical, almost bell-shaped.
Oh.
And when you combine that with its smooth, gelatinous surface, it creates this soft, pointed structure that, to Fries, closely mimicked the mouse ear shape of the forget-me-nots emerging leaves.
So he linked a fungal fruiting body to a plant's foliage based purely on, like, a fleeting developmental shape.
Essentially, yes. But there was also an ecological parallel driving his decision.
What do you mean?
Well, both the olive brownie mushroom and the water forget-me-not, specifically myosotis scorpioids, thrive in the exact same damp, marshy, waterlogged environments.
Also, they're neighbors.
Exactly, they are neighbors in the mud. So Fry's was attempting to link them by shape and by habitat. You have to remember, he was working without the benefit of modern genetics.
Still, it seems needlessly confusing to cross those streams, and clearly the scientific community agreed because looking at the literature since 1818, this fungus has been bounced around to an absurd number of genera.
Oh, it's been everywhere.
Right. The original genus Agaricus that Fry's used was basically just a dumping ground for any mushroom with gills.
Pretty much.
And as mycology matured, researchers kept trying to place it more accurately. It became Hypholoma myosotis, then Hypholoma myosotis, Naematolon, foliota, Naematolon.
And even Hemiphyoliota myosotis.
Has it a rap sheet of aliases.
Right, yeah.
I mean, in English, it's the olive brownie. In Welsh, the sources say it's Torth Widward, which translates to greenish-gray loaf.
Which is very descriptive.
Yeah. And in Russian, it's Hypholoma myosotis.
Oh.
Why has it been so impossible to pin down its actual evolutionary family?
Well, this identity crisis really highlights a fundamental tug-of-war in evolutionary biology. It's the battle between morphology, which is observable physical traits, and phylogenetic genetics.
Okay.
The confusion mostly centers on two major genera, Hypholoma and Foliota.
Let's break those down.
Sure. So traditionally, Hypholoma species have dry caps. They act as saprotrophs, decaying dead wood, and they drop these deep purple-brown spores.
We'll go foliota.
Foliota species typically have slimy or highly viscid caps. They also grow on wood, but they drop brown spores without any of those purple tones.
Okay, so the olive brownie must just break the rules of both clades then.
Exactly. It breaks all the rules. It has a highly viscid slimy cap, which suggests it belongs in Foliota.
Right.
But it grows terrestrially out of wet moss, not on dead wood logs like a typical Foliota.
And the spores.
Its spores are a dull, rusty brown, completely lacking the deep violaceous tones you'd expect to see in a true Hypholoma.
So it's a massive classification paradox.
It really is. In 1994, the mycologist Marcel Bon even proposed the genus Phenonemetalloma specifically to create a new category for mushrooms with viscid caps and brown spores.
Just trying to give this one outlier a proper home.
Exactly. But modern DNA sequencing has only deepened the mystery.
Wait, really? Genetics didn't solve it?
Not entirely. Genetic testing reveals it sits on this very isolated, specialized evolutionary branch. Sequencing of specimens in the Pacific Northwest recently uncovered a sister species.
What's it called?
Well, it currently holds the placeholder name Phenonemetalloma PNW01.
Catchy.
Very. But it shares the stiff stem and olive hues, but features olive yellow gills and a scaly stipe.
So the genetics are essentially telling us that this is a highly distinct ancient lineage. It hasn't fit neatly into our taxonomic boxes because it has been forced to evolve a highly specific physical architecture.
To survive an environment that kills almost everything else.
Right. It doesn't look like a standard forest mushroom because it doesn't live in a standard forest. So let's look at the mechanics of that physical architecture. I mean, how does a fungus engineer itself to survive a saturated bog?
It's actually a marvel of biological engineering. We can start from the canopy and work our way down.
Let's do it.
The palleus, or cap, is relatively modest. It measures between 10 to 40 millimeters across.
So pretty small.
Yeah, fairly small. The pigmentation is this complex mix of olive brown, bronze, and occasionally olive green. But the structural secret is its texture.
The sliminess.
Exactly. The cap is enveloped in a highly viscid, glutinous, gelatinous pellicle.
Let's define the physics of that pellicle for a second. Because in a humid, dripping environment, a simple, fleshy cap would just act like a sponge.
It could soak everything up.
Right. It would absorb ambient water, become completely waterlogged, and probably collapse under its own weight before the spores could even mature. So is this gelatinous layer acting as a hydrophobic barrier?
It functions exactly like a specialized biological raincoat.
A raincoat.
Yeah, and the pellicle is so distinct and robust that during fresh, humid weather, a researcher can physically peel this translucent gelatinous film right off the cap.
Wow, just peel it off in one piece.
Pretty much. And it shields the delicate spore-producing tissues underneath from ambient saturation.
That is brilliant.
Below that protective canopy, we find the lamellae, or gills. They attach squarely to the stem, which is a formation known as adnate.
And what do they look like?
They start as a pale cream color, mature through olivaceous tones, and finish as a dull brown. And the edges of these gills are finely denticulate.
Denticulate meaning?
Meaning they look under a hand lens, like a row of microscopic white teeth.
Okay, so the cap is brilliantly adapted, but the stem is where the proportions get truly wild in the sources.
Oh, the stem is ridiculous.
Yeah, because the cap is at most 40 millimeters wide, but the stem can reach heights of up to 150 millimeters while only being like 2 to 7 millimeters thick.
It's built like a needle-thin skyscraper.
Exactly. And thinking about the aerodynamics of spore dispersal, this makes total sense. I mean, if a mushroom in a bog had a short, stubby stem, it would drop its spores directly into the dense, wet boundary layer of the surrounding sphagnum moss.
And there is almost zero air current down inside that moss matrix.
Right. The spores would just sink into the wetness and rot. They wouldn't go anywhere.
So the entire architecture is designed to overcome that specific aerodynamic hurdle. The stalk, or stipe, exists to elevate those gills above the saturated boundary layer of the moss canopy.
Just trying to get high enough to let the released spores catch the microcurrents of wind moving across the surface of the bog. But to support a skyscraper that tall and thin in squishy, unstable peat muck, it has to have a serious foundation.
It does. The literature notes it features a deeply rooting base, anchoring itself far down into the dense layers of the moss to prevent toppling over.
And the surface of the stem is visually fascinating too, and has this rigid brownish base color, but it's overlaid with a pattern of silvery white veil fibrils.
Right.
They form these broken diagonal bands across the stem, creating the kind of snakeskin or zigzag pattern.
And that snakeskin pattern is actually a physical record of its rapid development. The olive brownie undergoes what mycologists call monovalangiocarpic development.
Okay, let's break down the mechanics of that. What does that mean?
So the primordial mushroom, the tiny little pin just starting to form, is entirely encased in a single outer universal veil.
So there is no secondary inner veil protecting just the gills.
Exactly. Just one outer layer.
Hmm.
Now, as that massive stalk rapidly elongates to push the cap skyward to catch the wind, that outer veil is subjected to immense longitudinal tension.
Just getting stretched and stretched.
Yes. The tissue is stretched to its absolute structural limit until it finally ruptures.
Oh, so the silvery zigzag bands left behind on the mature stem, they are the physical remnants of that ruptured veil.
Exactly. They are basically the stretch marks of its aggressive vertical growth phase.
That's incredible. To see the physical forces of growth permanently etched into the organism's skin like stretch marks.
It's a great visual.
So moving from the macroengineering to the microscopic, the literature points out some wild cellular features too.
Very wild.
The spores themselves are massive compared to related species. They measure roughly 13.6 to 18.4 micrometers long, and they are smooth but feature a highly prominent large germ pore at the distal end.
And that germ pore is a critical functional adaptation.
How so?
You can visualize it as a specialized, thinned-out region of the heavy spore wall. In H. myosotis, this pore is so large that the end of the spore appears truncate.
Like it's been cleanly clipped off.
Exactly.
I guess if the spore is armored with a super thick wall to survive the freezing acidic conditions of the bog, it needs a weak spot to break out of when it's time to actually grow.
The germ pore acts as a pressure release valve. When the environmental conditions are finally right, the right moisture, the right temperature, the internal pressure of the spore increases. The new fungal hyphae just burst right through that specific thinned pore to begin colonizing the peat.
Like an escape hatch. That's amazing. But the microscopy also reveals another vital cellular structure, right? The Chrysocystidia.
Yes, the Chrysocystidia. These are highly specialized sterile cells interspersed along the gills. They sit right among the spore-producing basidia, which are the microscopic club-shaped structures that physically bear the spores.
So what makes a sterile cell so important to taxonomists? If it doesn't make spores, why do we care?
It comes down to chemical reactivity. When a mycologist takes a microslice of the gill tissue, mounts it on a glass slide, and introduces an alkaline solution like potassium hydroxide KOH, a really dramatic chemical reaction occurs within these specific cells.
What happened?
An amorphous inclusion within the Chrysocystidium reacts with the alkali to form a highly refractive yellow-gold body.
Oh, wow.
Yeah, and the prefix chryso is actually derived from the Greek for gold.
So even when the macroscopic features are completely deceiving, like when the slimy cap mimics Foliota or the brown spores defy Hypholoma, a researcher can just drop a chemical on the gill and look for these hidden gold-flecked cells.
Exactly. That specific cellular chemistry is the ultimate fingerprint. It proves its true evolutionary lineage within the Strophareaceae family.
The microscopic chemistry just cuts right through 2 centuries of morphological confusion.
It really does. But, the chemical reactivity isn't just a convenient tool for humans with microscopes.
No.
No, the baseline chemistry of this fungus is what actually allows it to survive.
Right, because we have established the structural defenses. A tall aerodynamic stalk, a hydrophobic raincoat, a deep anchor. But structural engineering isn't enough to survive the actual ambient chemistry of a high-latitude bog.
Definitely not. The extreme acidity and the plunging temperatures require actual metabolic armor.
Metabolic armor.
Yeah.
Let's get into the secondary metabolism of the olive brownie, because this is where the true survival mechanics lie.
These aren't just structural tissues, right? They are highly complex biochemical factories.
Let's start with the pigmentation, because the olive, bronze, and yellow-green hues aren't arbitrary.
No, they're produced by complex polyketide pigments.
And polyketides are like this massive class of secondary metabolites, right?
Mm-hmm.
The literature specifies a compound called Naematolon, which drives the yellow and greenish-yellow tints.
Along with terphenylquinones, which are derivatives of polyporic acid.
It's a mouthful. But these pigments serve biological functions far beyond just coloration.
Oh, absolutely. Fungal polyketides in this family have been extensively studied for their antioxidant properties.
Right.
Specifically, lab tests demonstrate they exhibit incredibly high DPPH radical scavenging activity,
How high?
often neutralizing up to 83% of radicals at concentrations of 1,000 milligrams per milliliter.
Okay, let's translate that mechanism out of the lab for a second. A free radical is essentially an atom or molecule missing an electron.
Yes.
It's highly unstable.
Yeah.
If left unchecked, it will tear through a cell, aggressively stripping electrons off cellular membranes, proteins, and even DNA strands just to stabilize itself.
Which causes catastrophic cellular damage.
And that process is the definition of oxidative stress.
Exactly. And bogs are incredibly harsh, high-stress environments. You have the extreme acidity of the water, the intense UV exposure in open, treeless fens, and the drastic fluctuations in temperature and water levels.
And all of that generates a relentless barrage of these destructive free radicals within the fungal cells.
Yes. So the DPPH test, where researchers introduce a specific radical to a solution and measure how much gets neutralized, proves that the olive brownie is using these polyketide pigments as sacrificial electron donors.
Sacrificial donors.
Right. The pigment molecule willingly hands over an electron to the free radical. This neutralizes the threat before it can shatter the fungus's DNA, all without the pigment becoming unstable itself.
That is, it's literally a chemical shield.
A vital chemical shield.
But the sources also highlight a much more complicated class of compounds in its arsenal. Fasciculols, which are complex triterpene esters.
Yes, this is the part of the research that raised some serious red flags.
Because fasciculol is the exact same class of toxic compound that makes the olive brownie's famous cousin, the sulfur-toughed mushroom Hypholoma fascicular, violently toxic, intensely bitter, and completely inedible.
Right. The sulfur tuft uses fasciculol to cause severe gastrointestinal distress in anything that tries to eat it. It's a highly effective chemical defense mechanism against predation.
The extreme bitterness is a clear evolutionary warning sign. Yet, here's the paradox.
The tasting notes.
Yes. The literature states that while the olive brownie contains the exact same fasciculol toxins, foragers and field mycologists consistently report that H. myosotis has a mild taste.
Occasionally slightly bitter, but mostly mild.
So if it carries the genetic recipe for the exact same poison that makes its cousin notoriously bitter, why does it taste mild? Is it just hiding the poison better?
The current consensus points to the concept of a metabolic budget. We really have to examine the dietary differences between the two species.
Okay, let's look at their diets.
The sulfur cuft is a robust wood rotter. It colonizes dense, nutrient-rich dead logs, so it has access to a massive reserve of complex carbohydrates and lignin.
That gives it a lot of energy.
Exactly. It has the high-calorie metabolic energy required to synthesize and pump out massive quantities of these complex triterpene toxins.
So it has a rich diet, so it can afford the metabolic cost of building a massive toxic defense arsenal.
Precisely. The olive brownie, conversely, lives in an oligotrophic environment.
Write that down.
Oligo meaning few, and trophic meaning feeding. It is trying to extract nutrients from highly degraded, acidic, nutrient-poor peat.
It is essentially living on a starvation diet.
Exactly. It simply does not have the metabolic luxury to manufacture complex fasciculol toxins in massive concentrations.
Oh, that makes so much sense.
The genetic pathway to produce the toxin is intact, and it does synthesize it, but at a heavily reduced concentration due to its limited energy budget.
It knows how to make the poison, it just doesn't have enough calories in the pantry to brew a highly concentrated batch.
That's a great way to put it.
Though it is crucial to emphasize for anyone listening, a mild taste does not equate to edibility. The presence of these triterpene esters, even in lower concentrations, means the olive brownie is of negligible culinary interest and should be treated as toxic.
Absolutely. It is certainly not something to forage for the table. Its biochemistry is strictly dedicated to its own survival in the bog, not our dinner.
Right. And beyond the pigments and toxins, the cellular membranes of the mushroom are heavily fortified with specific lipids.
Yes. Analysis shows high concentrations of stearic acid, palmitic acid, and oleic acid.
Alongside ergosterol, which is a biological precursor to vitamin D. But let's consider the physics of a cell membrane in a freezing environment for a second.
Okay.
The cell membrane is a phospholipid bilayer. In moderate temperatures, it remains fluid, allowing proteins to move and nutrients to pass through.
Right. Nice and flexible.
But alpine and Arctic bogs experience brutal temperature swings. They routinely drop far below freezing. If a standard cell membrane gets too cold, the lipids pack tightly together, crystallize, and become rigid.
And then the membrane shatters and the cell dies.
Exactly. So how does it survive that?
To prevent that catastrophic crystallization, the fungus dynamically alters its own membrane composition.
Using those lipids we just listed.
Yes. Those specific unsaturated fatty acids, like oleic acid, have physical kinks in their molecular carbon chains.
So by pumping its cell walls full of these kinked fatty acids, it prevents the lipid molecules from packing tightly together when the temperature drops.
The membrane retains its fluidity and flexibility even when the surrounding bog water freezes solid.
It's synthesizing its own internal antifreeze.
It really is a masterclass in biochemical adaptation. Think about it. It wears a hydrophobic raincoat, it packs its cells with antifreeze lipids, and it pumps out electron-donating antioxidants to repair severe radiation and acid stress.
Which brings us to the most profound realization of the research, I think. This unique chemical diet of degraded, nutrient-poor peat isn't just about the fungus surviving.
No, it's much bigger than that.
The olive brownie's ability to eat the bog is what makes it a keystone species. It doesn't just endure the ecosystem, it is actively regulating it.
We are definitely moving from the survival mechanics of a single mushroom to the functional mechanics of the global carbon cycle.
Let's get into it, because Hypholoma myosotis is a highly specialized ecological operator.
And its territory is vast, yet incredibly fragmented. It has what we call a circumboreal distribution, meaning it circles the northern hemisphere.
Where specifically?
It's documented in the high bogs of Russia near St. Petersburg, across the peatlands of Canada's Atlantic Maritime Ecozone and British Columbia, throughout the U.S. Pacific Northwest, across northern Europe, and even isolated in the Australian alpine bogs.
So it spans the globe, but only exists in hyperspecific pockets.
Yes, it is restricted almost exclusively to oligotrophic peat bogs, fens, and the saturated margins of cold conifer swamps.
And its life cycle is inextricably biologically tethered to sphagnum moss and polytrichum moss. It fruits directly out of the living and dead moss layers.
And you have to realize, sphagnum moss is not a passive plant.
Oh no, it's a bully.
It really is an aggressive ecosystem engineer. Sphagnum actively alters the chemistry of the water around it to make the environment hostile to competing plants and microbes.
Let's delve into the mechanics of what the sources call the enzymic latch.
The enzymic latch, yeah.
Because in a typical deciduous forest, when a leaf falls to the ground, a massive community of saprotrophic bacteria and fungi immediately go to work.
Right.
They exude digestive enzymes that break down the cellulose and lignin, rotting the leaf and cycling the carbon and nitrogen back into the soil network.
But sphagnum short-circuits that entire cycle.
Wow.
The moss continuously releases complex phenolic compounds, including a unique polysaccharide network known as sphagnum, into the surrounding water.
Okay.
Simultaneously, the moss actively sequesters vital nutrients like calcium out of the environment.
So it's hoarding nutrients and poisoning the water.
Yeah.
This combination of severe acidity, Missing calcium and toxic phenolics just completely paralyzes the decomposing enzymes of standard bacteria and fungi.
The latch is thrown closed.
And what's the result of that?
Dead moss, dead insects, and organic matter simply don't rot. They just pile up in the cold anaerobic water, layer upon layer, slowly compressing over thousands of years.
And this is how peat is created. It is the reason we pull perfectly preserved, thousands-of-year-old bog mummies like the Tullen Man out of peatlands.
Exactly. The bog is a massive, permanent storage vault hoarding the world's carbon.
But H. myosotis is one of the extremely rare macroscopic organisms equipped with the highly specialized enzymatic toolkit required to bypass that chemical latch.
While the rest of the microbial world is paralyzed by the phenolics and the acidity, the Olive Brownie's enzymes are specifically adapted to function in those exact conditions.
They can actively digest this recalcitrant, stubborn organic matter.
It holds the chemical key to the vault.
It acts as a crucial recycler, slowly dismantling the dead moss and cycling a portion of that locked carbon back into the localized food web. That supports the microfauna and insect life that managed to live in the bog.
But its relationship with the bog's decay cycle presents a really brilliant ecological paradox.
The paradox being that while the olive brownie requires the baseline saturation and acidity of the bog to establish its mycelial network, fungi are fundamentally aerobic organisms.
Right, they rely on oxygen for cellular respiration.
And this perfectly explains a fascinating anomaly documented in the research from Romania, in the Panasi peat bog.
Oh, the drainage canals.
Yes. Field researchers discovered the olive brownie aggressively thriving, not deep in the undisturbed moss, but colonizing the exposed walls of man-made drainage canals.
Humans had cut deep trenches into the bog to drain the water table.
And this supposed bog lover was booming in massive numbers on the sheer walls of these artificial ditches. Why?
Well, the introduction of the drainage canal fundamentally altered the chemistry of the peat. By cutting a trench and dropping the water level, thousands-of-year-old layers of compressed peat that have been submerged in anaerobic, oxygen-starved water were suddenly exposed to the open air.
And that sudden influx of oxygen acts as a massive metabolic trigger.
Exactly. The olive brownie behaves as a pioneer of decay. It rapidly colonizes these newly aerated, disturbed zones because the oxygen allows its specialized digestive enzymes to work in absolute overdrive.
It is feasting on the newly accessible ancient carbon.
It is, and this specific behavioral trait transforms the mushroom into a vital bioindicator for conservation ecologists.
Because if a survey team is evaluating a supposedly pristine bog and they discover a massive, booming population of Hypholoma myosotis, it is not necessarily a sign of robust ecological health.
No, it's the opposite. It's a distress signal.
A distress signal.
A population explosion indicates that the bog is likely drying out, the water table is dropping, and previously locked peat is beginning to rapidly oxidize and degrade.
And we see this reflected in the data surrounding fungal membrane fatty acids, or MFAs.
Right.
When a bog is aggressively drained for agriculture or peat harvesting, the specific MFA profiles just plummet, indicating a catastrophic collapse of the specialized fungal community.
And even if conservationists step in and mechanically re-wet the bog by blocking the drainage ditches, the research shows it takes many years for the baseline fungal community and the delicate carbon balance to stabilize and restore itself.
These habitats are incredibly ancient and fragile. I mean, the literature references ecosystems like the Caledonia fen in the Australian alpine region.
Yes, where researchers pull core samples holding undisturbed pollen records dating back 140,000 years.
140,000 years. So being able to read the fungal landscape, knowing exactly what the presence of a specific pioneer fungus signifies, is essential for scientists attempting to preserve these ancient carbon sinks.
And because it serves as such a critical ecological indicator, accurate identification is paramount. A conservationist absolutely cannot afford to confuse the olive brownie with other species sharing the exact same habitat.
Which brings us to the complex world of comparative mycology and the doppelgangers of the bog.
The lookalikes.
Let's walk through the mental field guide required to separate the true sentinel from the imposters. The first major lookalike a researcher might encounter in the moss is Hypholoma elongatum.
Right, it shares the exact same sphagnum habitat.
So how do the evolutionary traits diverge?
While they share the habitat, H. elongatum exhibits distinct macroscopic and microscopic adaptations. Macroscopically, it is generally a more diminutive mushroom, and its cap pigmentation leans toward pale yellow or honey hues.
So entirely lacking the rich olive and bronze complexes of myosotis.
Exactly. And under the microscope, the differences are even starker. The spores of H. elongatum are significantly smaller, measuring only 9 to 11 micrometers.
More importantly, they lack the prominent truncate germ pore.
Yes.
So if they are living in the same harsh bog, why does elongatum produce smaller spores without that massive escape hatch we talked about earlier?
It likely points to subtle microhabitat exploitation.
Meaning what?
A bog isn't just a uniform flat surface. It is a highly textured landscape of raised hummocks and low-saturated hollows. H. elongatum may be adapted to fruit in slightly different microclimates within the moss matrix.
Requiring different spore aerodynamics and germination pressures.
Right, and that microhabitat concept perfectly explains the next imposter, Bogbodia uda, formerly classified as Hypholoma udum.
Bogbodia is a tricky imposter because it produces massive spores, similar to the olive brownie.
It does, but the critical difference is the cap texture.
The cap of Bogbodia uda is completely dry, or at most, slightly greasy.
Yes, it never develops the thick, peelable glutinous pellicle. It also lacks the stretch mark veil remnants on the stem.
And the absence of that gelatinous raincoat is a massive evolutionary clue, right?
Definitely. It suggests Bogbodia uda targets the drier peaks of the sphagnum hummocks.
So, by positioning itself just a few inches higher above the water table, the ambient humidity drops slightly.
Which removes the evolutionary pressure to expend energy, synthesizing a heavy hydrophobic gel layer.
It's incredible how a difference of literally a few vertical inches in a bog dictates entirely different biological engineering.
That's fascinating.
Now, the next look-alike on the list has a truly wild ecological backstory, Hypholoma tuberosum.
This one is great.
It's a visual imposter with an orange-brown cap, but its defining feature is hidden deep in the substrate.
If a researcher carefully excavates the stipe of H. tuberosum, they will find it attaches to a large, dense, walnut-like subterranean structure known as a sclerotium.
What exactly is a sclerotium?
A sclerotium is a hardened mass of dormant mycelial tissue. It essentially acts as a massive energy storage vault.
Why do they make them?
Fungi produce them to survive periods of extreme environmental stress, like severe drought or freezing. It allows them to remain completely dormant until conditions improve.
So the presence of a sclerotium is a profound survival mechanism. But the story of its distribution is what makes H. tuberosum famous in the literature.
Yes, because the species is originally native to the specific soils of Australia.
Yet in recent decades, mycologists have documented it fruiting globally, appearing in manicured gardens, potted plants, and municipal wood chips across Europe and North America.
And researchers traced this massive geographical leap back to a single vector.
The global peat trade.
Exactly. The fungus had heavily colonized commercial peat reserves in Australia. A single nursery in Sydney was harvesting this peat and utilizing it in commercial potting soils that were shipped internationally.
So the sclerotia, those hardened, dormant nutrient balls, simply hitched a ride in the soil bags.
Right. Surviving the dark, dry journey across the ocean, only to reactivate and fruit when a gardener in Europe watered their new plant.
It is a stunning example of how globalized commercial trade networks have inadvertently scrambled the geographical distribution of these highly specialized fungi.
It really is.
But returning to our native bog surveys, the final significant look-alike to rule out is Psilocybe olivaceo-tincta.
Yes, the Psilocybe genus is widely recognized because it contains the well-known magic mushrooms.
And the specific epithet olivaceo-tincta indicates it shares the olive cap hues of our olive brownie.
It does possess deceptive coloration. However, morphological differentiation is straightforward when observing the mature gills.
What happens to the gills?
They develop a dark purplish-brown tone, which contrasts sharply with the dull brown of myosotis.
And microscopically,
P. olivaceo-tincta entirely lacks Chrysocystidia. There are no gold-flecked cells reacting to potassium hydroxide on its gills.
so a trained conservationist uses the slime layer, the stalk remnants, and the hidden cellular gold to confirm the identity. But the literature details one final diagnostic phenomenon that sounds like absolute science fiction. It involves something called the UV glow quest.
Biofluorescence in fungi is rapidly expanding as a frontier of mycological research.
The studies note that the closely related sister species we mentioned earlier, Hypholoma PNW03, features gills that emit a bright, distinct glow when exposed to ultraviolet light.
Yes.
And because the olive brownie shares a highly similar complex of olivaceous polyketide pigments, it has become a prime target for researchers testing for biofluorescence.
which means there are mycologists systematically hiking through freezing, unstable peat bogs in the dead of night.
Just sweeping the moss with high-powered UV blacklights.
It's true. Field surveys utilizing ultraviolet illumination are yielding incredible discoveries across multiple fungal genera.
However, the presence of biofluorescence, or true bioluminescence, raises a profound evolutionary question.
It does. What biological purpose does emitting light serve for a fungus growing in an isolated bog?
We understand the chemical mechanics in some species. For instance, the compound hispidin functions as a key precursor in the enzymatic cycle that generates bioluminescence in certain fungi.
Right.
But the why is still heavily debated. What is the ecological utility of a UV glow for the olive brownie?
There are two dominant theories right now. The first posits that the fluorescence is entirely non-functional.
So just an accident?
Basically,
Yeah.
it may simply be an accidental chemical byproduct. We discussed the powerful polyketide antioxidants earlier.
The chemical shield.
Right. The complex molecular ring structures of those specific pigments may just inherently absorb invisible UV radiation and physically re-emit it as visible light.
Oh, I see. In this scenario, the glow serves no ecological purpose. It is merely a chemical quirk of its radiation armor.
A completely accidental light show.
But the alternative theory is much more interactive.
Much more. The alternative theory suggests that fluorescence is highly intentional and functional.
How would it function?
Well, in the dim light of dusk or the dark of night, a UV reactive glow could act as a specific visual beacon. It could be designed to attract nocturnal or low-light navigating insects native to the bog ecosystem.
Oh. So if a specific species of fungus gnat or bog meal is drawn to the fluorescent signal, it lands on the gills to investigate.
Exactly. And in doing so, it inadvertently covers his body in those massive truncate spores.
And when the insect inevitably flies off to a new, isolated patch of moss, it acts as a localized, highly targeted courier for the mushroom's reproduction.
Bypassing the need for wind entirely.
It is a brilliant theoretical mechanism, and it highlights a crucial point.
Yeah.
Mycology is not a settled science.
Not at all. The chemical networks and interspecies relationships operating in these extreme environments remain deeply mysterious and are actively being decoded.
There's an entire invisible world of chemical signaling and localized evolutionary warfare happening right beneath our feet, completely out of sight.
It's constantly happening.
I think that is the perfect point to synthesize everything we've explored today. We have tracked the olive brownie through a remarkable journey.
A very strange journey.
We started with an organism entangled in a 200-year taxonomic identity crisis, bearing a name confusingly linked to a rodent's ear and a blue flower.
We examined its masterful architectural engineering, the towering aerodynamic stalk marked by the stretch marks of its rapid vertical growth.
And the hydrophobic, gelatinous pellicle shielding its spores from the saturated decay of the bog.
We detailed its highly complex secondary metabolism, too. It survives freezing temperatures by altering its cellular membranes with kinked, unsaturated lipids.
And it defends its DNA from radical oxidative stress by deploying a localized arsenal of polyketide antioxidants, all while managing a low-concentration toxic defense on a starvation diet.
And most critically, we establish its role as a master of the peat.
Yes, it possesses the rare enzymatic ability to bypass the sphagnum moss's chemical latch, recycling ancient organic matter.
And serving as a vital bioindicator for the health of our planet's most ancient and fragile wetland ecosystems.
It is an organism that perfectly bridges the microscopic nuances of cellular chemistry with the macromechanics of the global carbon cycle.
It absolutely does.
Which brings me to a final lingering thought I want to leave you with.
Let's hear it.
We have established that sphagnum bogs are the Earth's ultimate carbon vaults. They hoard up to 140,000 years' worth of undecomposed organic matter in a cold, acidic, oxygen-starved suspension.
Right.
And we know that when those vaults are disturbed, like when human drainage canals introduce oxygen into the deep peat fungal pioneers like the olive brownie immediately rush in.
Because the oxygen supercharges their enzymes.
Right, and they aggressively feast on the ancient carbon, rapidly breaking it down.
The metabolic restraints are totally lifted, and the decomposition cycle accelerates exponentially.
Exactly. So here is the provocative reality we are currently facing. As anthropogenic climate change permanently warms the globe and fundamentally alters historical precipitation patterns, these ancient bogs are beginning to dry out on their own.
The water tables are dropping globally.
Yes. So as the global water tables drop and millions of acres of deep peat are suddenly exposed to atmospheric oxygen, what happens to that baseline fungal community?
It's a huge question.
Could these microscopic fungal pioneers, in their natural localized evolutionary rush to consume this newly oxygenated buffet, inadvertently act as a biological catalyst?
Wow.
By doing exactly what they evolved to do, could they aggressively accelerate the release of hundreds of thousands of years' worth of stored carbon directly back into our atmosphere?
It is a deeply sobering feedback loop that climate modelers and soil ecologists are racing to quantify.
The stakes are astronomical. So the next time you find yourself near a quiet, wet patch of moss, or you merely picture that freezing, inhospitable bog we started our journey in, remember the scale of what is occurring just out of sight.
There is a lot happening under the moss.
There is a microscopic, highly complex chemical war for the stability of the Earth's climate happening right beneath your feet. Thank you for joining us on this deep dive.